So this last one for the metabolism will be enzymes, and this is what our lab will be this week. So this one is going to be slightly longer. What I would like you to think of is all, see if this makes sense to you. All enzymes are proteins, but not all proteins are enzymes. Say that one more time. All enzymes are proteins, but not all proteins are enzymes. So I don't know if that makes sense to you. Hopefully it does. And then right after that, I want you to write down in your notes, that's bullshit. It turns out we thought that was true for years, but we now know it's not true. There are actually some RNA molecules that actually act as enzymes. But the reason I'm still giving that, this idea is because the enzymes we're going to talk about, and most of them are proteins. So that's really what we're going to be kind of mentioning throughout this is how those work. What you need to know is enzymes are biological catalysts. They speed up metabolic reaction by lowering the energy of activation, which we'll talk about. What I need you to understand is enzymes are not necessary for these reactions to go. Per se, I mean, these are reactions that would occur anyway, but what an enzyme will do is speed up that reaction, especially for living things. You know, I always use this example is, you know, we talk about enzymes and how they break down things. Like sucrose here, we have sucrase. Enzymes typically end in ASE. And so you can usually guess it's an enzyme because of that ending. But I want you to realize that sugar, sucrose, would break down on its own. But for us, it might not be fast enough. For instance, if you eat a big In-N-Out burger, you know, and have no enzymes, that burger is going to break down. But you don't want to be sitting around going, oh, I don't really feel well. That In-N-Out burger I ate 6 weeks ago is just kind of sitting there. So obviously we need it to happen in a time that is relevant for us. These exergonic reactions here, you can see we talked about those on an earlier thing, but what we're going to do here is we're releasing energy. And so here we have A and B, and we're going to kind of recombine these down here. But to do that, you have this little transition state. And this is the energy of activation, the amount, you know, that's necessary to get this to go from here to here. You know, what an enzyme actually does is has to deal with this energy of activation. So if you move over here and look at this picture that's been slightly modified, what you're seeing is that with an enzyme, that energy of activation is a little bit lower. What you have to realize is these are proteins, and proteins have those you know, that 4 levels of shape that we talked about. And so what's important is, you know, you change that shape a little bit, it may make things easier to go. For instance, you know, examples that I might do in class is like a pencil breaks. You know, to get a pencil to break, you got to put some force on it. But if you had something like a rubber band on it that was pulling it and bending it to start with, the pressure that you would have to put on that pencil to finish it off to break it would be a lot less. The same with, like, if this was a speed bump and your car's on a hill, if the speed bump is this high and your car is sitting behind it, the energy it would take for that car to roll down the hill would be quite a bit. But if that speed bump, you put an enzyme on it that lowered that energy, you know, that little speed bump, it would take less energy. Okay, so again, they don't change the reaction. They're still going to end up where they're going to end up. It's just about the speed it takes. All right, some other terms that we need you to know. Substrates typically are substrate specific. So that means they typically work on one thing. So sucrase works on sucrose. You know, if you're lactose intolerant, it's because you lack the enzyme lactase. So again, sucrose is broken up by sucrase, lactose, lactase. Starch is broken up by amylase. It's not starchase. Starch is actually a long polymer of glucose. And there are a couple of different formats. One is in the form of amylose, and so breaks up the amylase. Here's the part I need you to understand. So you have this globular protein, that 3D, third level structure of a protein. Within that, typically you have an active site. You have this area, and here you can see on the bottom picture here, this little open area down here, this little pocket. And what happens is the substrate, anything, when we say substrate, that's what the enzyme works on. So the substrate for sucrase is sucrose. The substrate for lactase is lactose. So anyway, so lactose is coming in, it goes into the active site and combines and forms a enzyme substrate complex. What's happening is you've got to realize in the active site, you have the amino acids, and so they have the R chains, and you have these functional groups that are exposed. And so a particular substrate also will have functional groups that are exposed, and then you have some reaction. Once that thing enters, there's what's called an induced fit. And that induced fit is when the protein actually wraps around the substrate. It actually will change the shape of not only the protein, but even the substrate. So it's like me bending the pencil kind of thing. And so we want you to know about that induced fit. They say it fits like a lock and key, or sometimes it fits like a glove, not OJ's glove, if you remember that, but anyway, so that's that concept. And then what happens is once that reaction has occurred, the enzyme is recycled, and it could be used over and over again. So good stuff. So here's kind of the bigger picture, kind of walking you through. And here they have two substrates that you're trying to they're coming in, and then two products that are coming out. So a little bit different than the single one I was talking about, but same kind of concept where you have this complex formed, an induced fit, which is a little hard to see in this one. But you can see it, the shape here is maybe a little bit different. And then this thing is put out again. And you can see kind of a bending going on here. So I guess that's talking about that induced fit. All right, for enzymes to work real well, they have cofactors and coenzymes. I want you to kind of know the definitions. It helps the induced fit. So a cofactor is a non-protein helper, an inorganic helper. Typically, that's why we need vitamins and minerals. Those things are actually act as our non-protein helpers. Then coenzymes are actually protein helpers. So there are other things that might be necessary for these to work properly. All right. The other thing I want you to realize is we have inhibitors. This is something we're going to talk about. You're going to see in lab. There are two types, competitive and non-competitive inhibitors. And so here's the typical active site. Here's your typical substrate. Things go in, things lock in, things are doing well. Competitive inhibitor would be something that actually blocks the active site. It does not allow the typical substrate to get in. To give you examples of that is the one example I typically give is RAID. A lot of insecticides actually act as competitive inhibitors. There is a protein in your body, in your nervous system, cholinesterase is an enzyme that actually cleans up neurotransmitters. And so what happens is you have these neurotransmitters that go from one neuron to another, and they sit there and they shoot the next neuron off. But then this enzyme comes in and clears it up to shut that down. What RAID will do is actually it will attach to those, not shut them down. And so that neuron would continue to fire over and over until it shorts out. So what you're really doing when you're using that stuff is you're shutting down the nervous system. Because you can't wait to see the ants die. You're spraying way too much on there and probably drowning them. But reality is same thing. And one of the reasons that insecticides are dangerous to us is the same thing. It does exactly the same thing in us. But because you're much larger than an ant, you don't necessarily shut down like that. And that's why they tell you not to use it in closed spaces and all kinds of different things like that. All right, a non-competitive inhibitor is actually one that binds somewhere else. So what happens is here's an inhibitor that hooks on somewhere else on the protein, and then what it does is it changes the shape of the active site enough where the substrate now does not fit properly. And these sites are called allosteric sites, which I have that term on another slide, which is right here. So the allosteric site will take non-competitive inhibitors. It can also take activators, things that will actually increase how fast an enzyme works. And then you have cooperativity. I just put this on here. It's not a concept we talk about a lot, but often what will happen is when you set one off, multiple ones will set off. It'll start a cascade to help things work a lot quicker. So so here you got 4 active sites. When you fix one of them, all 4 of them kind of go into usage. So the idea is that it's not like you have to hit the switch for each single one. One switch will turn everything on. And this I'm not going to hold you to, other than I want you to realize the way we are working is that we have all these different intermediate steps with enzymes, and there's typically a feedback mechanism involved that will shut things down. So your enzyme's breaking something down, and you get to your product. Once you get enough product, that will come back up here, slip into maybe that allosteric site or even the active site, but usually the allosteric site. And it'll shut this thing down. It's like, let's not continue to do this. And then once you run up and use up this product, that'll slip out, and then all of a sudden the process will run again. This is called a feedback inhibition loop. We're going to talk about these a little bit more when we talk about some DNA stuff too, but just wanted to give you kind of that concept. And so that will end the